Good afternoon, everyone. My name is Brad Johnson, and it is my great privilege to serve as Dean of the College of Science and Engineering here at Western Washington University. And on behalf of the College of Science and Engineering, the Graduate School, Northwest Pathology, and the WWU Alumni Association, I thank you for attending today, and I'm very excited to welcome you to today's webinar, Pandemic Partnership, WWU and Northwest Pathology. And now to get things started, I'm going to introduce biology professor, Dr. Nick Galati.
Thank you for the introduction, Brad. So thank you all for coming today. Our panelists for today consist of Dr. Gregory Wolgamott from Northwest Pathology, biology professor Anu Singh Khande, two WWU graduate students, Hannah Fisher and Benjamin Hagan, and two recent WWU alums, Aliki Valdez and Carrie Roberts Nygren. And so now we're going to let every one of our panelists do a quick introduction. So Dr. Wolgamott, why don't you begin?
Thank you very much. My name is Greg Wolgamott, and I'm one of the pathologists here in Bellingham and one of the directors of Northwest Laboratory. And I did my medical training at University of Washington at Harvard, and then also did a PhD in molecular biology at the Fred Hutchinson Cancer Research Center. Whereas not really directly studying cancer, but actually worked on identifying and cloning a novel retrovirus at the time. And so currently I'm one of the directors of the laboratory and have had the privilege of working with many talented people in this partnership to provide COVID testing to the community and to many other states as well.
Thank you. How about we move on to Hannah next?
Hi, I'm Hannah Fisher. I did my undergraduate at Western, and I'm now a current second-year graduate student at Western's graduate program, and I work here at Northwest Pathology as a molecular supervisor.
Thank you, Hannah. Benjamin?
Hi, my name is Benjamin Hagan. I did my undergraduate at University of Washington, studying cell and molecular biology and evolution, and doing some research in plant biology. I'm a second-year grad student at the Western Washington, studying under Dr. Dan Pollard, and I'm a PCR technician at Northwest Labs that joined last March.
Thank you, Benjamin. How about we move on to Aliki?
Hi, my name is Aliki Valdez. I graduated from Western in June of 2020 with my molecular and cellular biology degree. During my time at NWP, I worked as an RNA extraction technician.
Thanks, Aliki. And how about we wrap up with Carrie?
Hi, I'm Carrie. I went to Western for molecular and cell biology. I graduated a year ago, and now I'm a PCR tech here at Northwest Pathology. Been here for a little over 9 months.
Thank you, Carrie. So now let's take care of a little bit of housekeeping before we dive into the questions. So some of you have submitted questions along with your registration, and we really appreciate those. And now as we move through the discussion today, if you have additional questions, please use the Zoom Q&A feature at the bottom of your screen. There should be a button at the bottom middle of your screen specifically, and you can send us any kind of off-the-cuff questions that you have as we go through the discussion today. We'll be responding to those questions, and we'll get to as many as we possibly can. And now, before we get started, I'd also like to just take 1 second to acknowledge that there's many other students across the Western campus that have played a really important role at Northwest Pathology, and they come from across campus, multiple departments, and so kind of sad that we can't have everybody here, but we really appreciate all the support that we've had from across campus. Okay, so let's get started with a question for Dr. Wolgamott. I'd like you to be able to give us an overview of the context. Specifically, what led to your decision to get testing for COVID-19 up and running at Northwest Labs?
Yeah, it's a good question. And really the decision to get the testing, I mean, there was really no other choice. And so if you can just remember what it was like a year ago, you know, the pandemic was fresh, you know, schools were closing, people were scared. The hospital was drowning. And so, for example, at the time that we started the testing, there were 64 people in the hospital that were waiting for their tests. And on average, it was taking about 10 days to get a test result. The reason why I was taking it was so long is because at that time, so few labs were allowed to do the testing. And it was relatively late in the game that the FDA opened up and allowed other labs such as ours to begin testing too. So once they made that decision, really the next day, we made the decision that we had to bring it on. So with the hospital being full of people that they didn't know who had COVID, who didn't have COVID, that causes huge problems for the hospital because they can't figure out who to isolate, which parts of the hospital you put different people so you don't expose other vulnerable people to COVID. And so they almost have to operate under the assumption that everybody has COVID, and the amount of personal protective equipment that they were consuming in the hospital with N95 masks, gowns. I mean, they were just consuming all their stocks of PPE, which now is becoming national shortage. For example, then you couldn't order more N95 masks because they were all scooped up. It was also causing problems with discharging patients because any patients that could potentially have COVID could not be discharged back to their nursing homes, for example, because the nursing homes, those have some of the most vulnerable people in the entire community, and they wouldn't accept anyone back who may have COVID that could expose their other people. So then the hospital fills up, and then that starts a chain reaction that impacts other people. So the people that are having heart attacks, things like that, can't get into the hospital because there are literally no beds left. So, so a year ago, the situation was extreme. And so the decision to bring on the COVID testing, I mean, there was really no other choice. We had to do it because we knew that we could. And some of that story will evolve over this next hour on how that was accomplished.
Very good. Thank you, Greg. As a follow-up to that question, would you be able to kind of guide us through the effort and the development that it took to get Northwest Pathology over the hurdle and to allow them to actually undertake this major expansion in capacity?
Yeah, there are a lot of hurdles that needed to be crossed. The first was, of course, identifying which testing platform to bring it on and getting it validated. So we did that relatively early on. And then the remaining hurdles basically could be sorted into equipment and supplies, and then the most important one is people. As far as supplies goes, that includes not only the reagents for running the tests, which are now in critical demand, because now there's worldwide competition for them, but even simple things like swabs. If you remember, there was a lot of news stories that swabs, nobody had any swabs. And so, for example, to get swabs, we had to register as a Korean importer with South Korea, and we had, hundreds of thousands of swabs imported from South Korea. And so we had a lot of people on our team that were really ingenuitive to try to, source equipment and swabs. And then the final hurdle that is really mostly the focus of today's discussion is the people. Now, we already had a fine team of people in the lab that ran the core tests that we do, and they're super experienced, bright people. And early on, they were very helpful in getting the testing going. But it quickly became apparent that we needed a lot more people that were smart and that had training and understanding of molecular biology. And it's kind of an interesting story. I was out pacing around the neighborhood trying to think about how to solve this problem because we wanted to do the testing, but we needed more people to help. And I just happened to bump into Theyve Hooper, one of the professors at Western Washington University. And it suddenly occurred to me that I had heard in the news story that a bunch of the labs at the Western campus were closing because of the pandemic. And so I thought, hey, this is a great source of people that are knowledgeable and educated about this stuff. And so maybe we could bring some of them to join us to help with this endeavor. And so Theyve Hooper was super helpful, and he reached out to other professors and the students and the faculty. And very quickly, we had a whole bunch of people that wanted to take action and help out. And so early on, we had a name for the team that helps from Western Washington. We called them the ACT team, the Awesome COVID team, which is our mnemonic, which never really stuck. Maybe it's a little bit too cheesy or something. But that's really what we thought of them, is that here we are in a time of need, and we had a whole bunch of people that were interested and willing to help use their skills to help solve this problem.
Thank you, Greg. That's a really remarkable story. And now let's move on to a question for Dr. Singh Khande. And I want to first apologize because I was so excited to get into the webinar that I forgot to allow her to introduce herself. So why don't you introduce yourself and then we'll get into your question.
Right. Hello, everyone. My name is Anu Singh Khande. I teach in the biology department here at Western. I teach molecular biology classes, including a lab class called Methods in Molecular Biology. In this methods class, students receive hands-on training in molecular biology procedures, such as the PCR that you've already heard about. So students do lots and lots of PCR in this particular lab. So that's by way of introduction, Nick.
Perfect. And so that leads us right into the question. So can you describe a little bit about what you did with Northwest Pathology in order to help get the COVID testing up and running?
Yeah, so when the pandemic hit our shores, one of the trying things, at least for me, one of the many trying things, was this feeling of helplessness, the not being able to do something constructive. So when Theyve Hooper issued this call to arms that you heard Greg mention, I jumped at the chance and I joined the group, the awesome group, and participated in the testing. I did the qPCR test Nick, would you like me to run through some slides to explain that testing procedure at this point?
That would be fantastic. Thank you.
Okay, so I'll share my screen at this point. So let's take a look at the virus that causes COVID. There's genetic material on the inside, and the genetic material of this virus is RNA, which is a close cousin of DNA, the genetic material that's in all of our cells. Now, there's some proteins as well, like this N protein that you see on the inside. And then this whole kit and caboodle is wrapped in an oily layer, which has additional proteins, its own unique proteins, like this E protein and the M protein in the envelope, in this oily layer. And then there's the infamous spike protein. The spike protein is infamous because it's the chief means by which the virus enters the host cells, our cells, because it docks into a membrane receptor that's present on the surface of most of the cells in our body. And that's how it gains ingress. That's how it triggers a sequence of events that enables it to enter the into our cells. And now, how would we go about looking for the presence of this virus in a sample, in a potential patient sample? One means of detecting the virus is to look for the proteins themselves, the E protein, for example, or the N protein. And the rapid antigen test does exactly that. It looks for the presence of virus by looking for these proteins that are unique to the virus. Now, there's one downside to the rapid antigen test. It only works when there's lots of the virus within the body, around the peak of the infection cycle. So if I got infected yesterday, it's unlikely that the rapid test would detect the infection because there just wouldn't be enough of the protein for that test to pick up the virus. So the gold standard for sensing the presence of the virus, for detecting the presence of the virus, is this PCR-based test. So the PCR-based test begins with let's say a health professional collecting a sample from the nose and throat, or the mouth and throat. And so that's the swab that Dr. Greg was mentioning. We need these swabs to collect the sample. The sample is then stored in this special solution that preserves the sample and protects it. And then the sample arrives at the lab, and that's when there's extensive cataloging and barcoding to be sure that the sample and their identity can be tracked with great accuracy. And then begins the process where technicians, such as the folks you've heard from already, who extract the genetic material from the virus. So the virus is ripped open and the genetic material is extracted. This is actually these days done by a robot. And then the technicians use the genetic material of the virus to generate DNA. So the virus has RNA as a genetic material because and RNA is a close cousin of DNA. And so RNA is converted into DNA. And that's when this technique called PCR kicks in. So PCR is a technique that can take tiny amounts of DNA from one molecule of DNA, make many, many billions of molecules of DNA. So RNA is turned into DNA, and then the genetic material of the virus, now in the form of DNA, is amplified by this special machine. So as those billions of copies of the genetic material start to accumulate, the machine tracks it in real time. So we use a particular version of PCR that's known as quantitative PCR. So quantitative PCR actually allows you to follow the conversion of or the amplification of DNA in real time. And so if the viral genetic material is present, this is the genome of the virus and the many genes that it codes for. So we look for specific genes that are specific to this particular virus, the SARS-CoV-2 virus that's responsible for COVID-19. And so as the viral genetic material is amplified in the machine, we can see it in real time. And of course, if there's no viral material in the sample, then that amplification does not occur. And the machine keeps track of that and tells us exactly how much of the viral genetic material is accumulating. And that's why it's quantitative, because it actually tracks how much how much genetic material was present in the sample. If there was very little, we would know that. And if there was a very high viral load, lots and lots of the viral particles, then we'll see very much more accumulation of the genetic material. So that, in a nutshell, is how the PCR test that's quantitative works. The RT refers to this business of converting RNA into DNA. So that's a quick tour of how the test works. I'll stop sharing my slides now. Excuse me. All right, back to you, Nick.
Thank you so much for the outstanding introduction to the COVID testing. I mean, it's become such a household, idea at this point, and it's fascinating to hear how the test actually works at the molecular level. So now let's hear from some of our students. So we've got a question now for the undergraduates, the undergraduate alumni. So how did you start working at Northwest Labs on the COVID testing? And how did the biology program at Western specifically prepare you to work in this kind of new collaborative scientific environment? And Aliki, why don't you lead off?
Sure. So I was sent a job description that was circulating through the biology department, looking for people who were proficient in basic molecular biology techniques like preparing reagents, PCR, things like that. And I pretty much met all the qualifications that they were looking for through my experience at Western. My favorite things about the biology department at Western is just how welcoming and constructive the learning environment is. Having the opportunity to be vulnerable and ask what may seem like simple questions really allowed me to grow as a scientist, not to mention all the hands-on experience I was able to get from the lab classes offered. You're always taught from the perspective of, why are you doing this? Why does this matter? It's one thing to be told what to do versus actually understanding the reason behind why you're doing it. You don't realize how important and impactful the little things are until you're on your own. And so already being familiar with the equipment and even the methods really give you a boost in confidence when you first start off. And I was able to realize that once I started working at Northwest Pathology.
Thank you, Aliki. That was really informative. Carrie, do you want to chime in on this one? So how did you start working at Northwest Labs? And is there anything about the Western Biology Department that prepared you to work there?
Yeah, so I started working at Northwest just a little bit after I graduated. So many of my friends I made at Western were working there. So I heard about the job that way. And And Western had a huge hand in preparing me for this job, especially working in Dr. Suzanne Lee's lab. I did a lot of RT-PCR and picked up some really valuable wet lab skills, was really able to hit the ground running here. So, yeah.
Cool. And then I heard there's word on the street is that you're going to give us a little tour. Is that correct?
Yeah, Hannah and I are at work right now, so we will give you all a tour. Just going to go get Hannah. Hey, are you ready? Okay. Just gonna flip the camera. Okay. So we are walking through some offices right now. Walking through Core Lab, which is another part of Northwest Labs. And if you can't hear me, somebody let me know because there's machines going and stuff. So here's the COVID Center at Northwest Labs. There's Supervisor Joey. We have some samples shaking here. We have somebody working in the hoods. These are the robots that pipette the COVID samples into our extraction plates. These are Kingfishers. These are the machines that actually do the RNA extraction. These are our qPCR machines. And I have some results pulled up here. So this is what a positive result would look like. We have all 3 genes that we are amplifying present, and our internal control is also amplified. This is what a negative result looks like. So there's no amplification for those 3 COVID genes, but we still have our positive control. Yeah, this plate has quite a few positives on it, which is not great, but we see it all the time. Yeah, we have somebody doing PCR over here right now. Yeah, this is also, yeah, another Western alum. And this is where we set up the PCR reaction over here. Yeah, Hannah, add anything? Maybe we can show them the decapper robot. Yeah. Before we got the robots, this was all done by hand. And we still have a lot of amazing extraction techs that spend a lot of time uncapping things. But we do have these new Fluid X decappers. They take those orange tubes and they'll take the cap off for us. And then it can be loaded onto the Integra Assist robot here on this side. And we definitely appreciate it now because when we first assembled last March, we didn't have these. And so it's kind of cool to see how things have evolved over the last year. Yeah. Yeah, is there anything else that we should point out while we're here?
No, I mean, that's an amazing infrastructure, an amazing set of equipment. I mean, I never really appreciated the scale of what was going on there, and that's quite impressive.
Yeah.
Cool. So thank you so much, Carrie, for the tour, and Hannah, thank you for the tour. I think that was a really neat glimpse into what life is like working at Northwest Pathology. Very impressive. And now I was hoping that maybe we can move on to our graduate students and ask them a couple of questions. So Benjamin, to start, could you please provide us with a little bit of insight as to kind of how your experiences as a Western graduate student have enabled you to work at Northwest Pathology, and maybe how you became involved and what your role is at Northwest Path?
Sure. Yeah, definitely. Well, like I said in my intro, I'm a 2nd-year graduate student, and I joined Northwest Pathology close to a year ago when COVID testing was first getting off the ground. Thankfully, I had a little bit of lab experience before I was able to hop into doing COVID testing. And my experience was mainly centered around doing molecular techniques and doing PCRs, genetic testing, and which gave me a lot of good background knowledge to be able to understand the protocols that we were concerned with at Northwest Path. And thankfully, since I was doing a lot of research in a graduate research lab with Dr. Dan Pollard, I was able to have some experience troubleshooting protocols and have some experience really wrapping my head around the larger impacts and the larger biochemistry that happens with the specific tests that we might be running at Northwest Path. So I feel like that background and that experience being able to troubleshoot when things go wrong allowed me to assist getting the procedure that you were seeing during that tour a little bit off the ground. And that came through a lot of mentorship at both UW, where I did my undergraduate, and at Western, where I'm doing my graduate work, and a lot of collaboration with students that I've been working with for the last 2 years that are either working at Western doing their own research, or at both places doing clinical testing and Western research. And a lot of the experience that I'm getting at Northwest Path has also allowed me to be better, well-rounded as far as my skill set when I'm looking for jobs, and has allowed me to really develop some of my other experiments that might be relating to my thesis research. So it's really a two-way street. Both sides give me solid experience, and I've been very grateful. Right now, I am taking a little bit of time off from testing, but in normal times, I am a PCR technician. So I set up the PCR reactions where you amplify the RNA into DNA and really test for those COVID-specific genes using our primers there. And so when I'm in the lab, I'm doing a lot of pipetting, a lot of transferring samples to and from the different reactions and from the kingfisher that you saw in the video there.
Perfect. Thank you, Benjamin. Hannah, you were finishing up the tour, so let's move on to you next. Can you provide us a little bit of insight into your experience as a Western graduate student working at Northwest Pathology? And maybe could you tell us a little bit about how you became involved and what your current role is at Northwest Pathology?
Yeah, hi. So like Dr. Singh Khande said, when all of this was starting to kind of unravel last March, research labs were being closed at Western. We were moving to an online format. Many of us also serve as teaching assistants, so we were frantically getting our online platforms ready. And so it was just kind of a stressful, isolating time. And to get the email from Dr. Hooper and Dr. Walgamot, it was a really cool way for us to kind of do something since we couldn't be in our labs, and you know, it kind of felt helpless for a while. So it was nice to be able to do something in such a stressful time. And so, yeah, I was part of the original hire group last March. We just celebrated the lab's birthday the other day. And so, yeah, it was a cool experience to see it even before it was fully validated. I remember sitting in a conference call with the doctors and Thermo Fisher, and just like, it was just a really cool experience to see what it takes to get a clinical test off the ground. And so I started out as a PCR tech, and so doing what Benjamin mentioned, a lot of pipetting. All of us also worked as extraction techs. Depending on sample volume, we would jump in and do that. And then recently I've become a supervisor here at the lab. And that basically entails more of like oversight duties, such as just making sure things are running smooth. These guys don't really need it. They're great. We've got a really great team, but we help with that, and we help with generating like standard operating procedures, making sure things are up to CAP standards. There's a lot of things that have to be maintained so that we can keep testing going all over the place, not just in Washington. And I think probably my favorite part about this would be that we get to assist, and we also have extraction techs and PCR techs assist with this, but validating other assays or conditions that we might do testing. And so while we're also doing testing all the time, we're also looking to make our process better. How can we make testing more accessible through batch testing, things like that. So it's been a great experience. And all of the techniques that I've used here, Western really laid the foundation for that. I took Biology 324, which is the molecular methods class that Dr. Singh Khande mentioned. I also had the privilege of teaching it with her this last quarter as her teaching assistant. And that class really lays down the foundation. You learn how to do PCR. We talked about COVID testing so that our students could have an understanding of what's going on and how to jump into that if they needed to. And so, yeah, and I'm also very privileged to be in a research lab at Western. I'm in Dr. Galati's lab, and while we do a lot of quantitative microscopy work, I do get to do some PCR, and it all kind of blended together and helped support kind of my career goals going forward, mixing biology and medicine. So it's been a good ride.
Really nice, Hannah. Thank you. So I think now we've got some questions popping up from the audience. So maybe we can take a few moments to go over some of the questions from the audience. Let's see here. Here's a question about the specificity of the primers. So given the specificity of the primers that are required for PCR, is Northwest Pathology able to test specifically for any of the new variants of the virus that are emerging? And I guess that might be a great question for Dr. Wogelmont.
Yeah, that's an excellent question. And the answer is sort of. So the primers are very specific for this particular coronavirus. There are other coronaviruses that circulate as common colds, and there's 4 common ones that have been circulating in the community also. And then there's also like MERS and such like that have had sort of like mini epidemics in the past. And these primers do not cross-react with any of those. And so these only react with the SARS-CoV-2, the cause of COVID-19. Now, regarding variants, this is a big topic these days. And so one of the main variants, B117, people call it the UK variant, that has specific changes, particularly in the S gene, which encodes the spike protein. And a lot of times in viruses, that's where you see a lot of mutations and variants arise in the spike protein, because that's particularly important for immune responses. And that's, for example, why the vaccines are directed against spike proteins. And so that's a place where you tend to see a lot of mutations. Now, one good thing is the test that we run, we believe is one of the best tests out there, because in every test, we're actually looking for 3 different genes, as was already mentioned. And so we look at ORF1, we look at N, which is the nucleocapsid gene, and then the S, which is the spike protein. And so the UK variant, for example, it has this deletion that's called deletion 69-70, where there are 6 nucleotides deleted out that causes in the protein there to be 2 amino acids that are dropped out. And that overlaps with one of the primer binding sites in the S gene. And so for that variant, we do not see amplification of the S gene. So we call that an S dropout. Fortunately, because we also look at 2 other genes, we can still detect those viruses just fine. And furthermore, we can say, hey, this is probably a UK variant. Now, the deletion, for example, seeing S gene dropout, that alone does not define UK variant, because you have to look specifically like at a change at position 501 and such, that our assay doesn't give us any information about. But what this does is this gives us an enriched sample that we then send down to the University of Washington and Department of Health, and they can do the more detailed sequence, where they actually sequence the S gene and can tell us exactly what variant it was. Early on, we were seeing a lot of S dropouts that were not the UK variant, but it was another local variant of uncertain significance that was circulating. Since then, the UK variant has kind of taken over more. And so the majority of S dropouts that we're seeing are UK variants. And I believe about 10% of positives or so represent that now. Now there's other variants too. There's not just UK, but for example, there's the South Africa variant and a Brazilian variant, and neither of those have that deletion in the S gene. And so both of those just look like normal SARS-CoV-2 in our assay. We cannot detect those variants. I mean, we still detect them in the sense that we get a positive test, but we can't specifically say that they're the variants. And so really a lot of these variants, the only way you can detect those is actually by full sequencing of the S gene. That's how they're detected and classified. And so none of the high throughput assays can do that. And so as you probably know, it's a very labor intensive technique. And so the Departments of Health and the University of Washington are doing several hundred of those per day. So we're also just sending just regular old positives. Several hundred per week we send them in a sequence to look for the arrival of some of these other variants.
Okay, so it sounds like, you know, there's no direct ability to confirm at Northwest Pathology, but there's easy follow-up ways that we could start tracking some of the variants by sending out some samples.
That's right. Yeah, we're in direct partnership with the University of Washington Departments of Health, giving them lots of positives that they're seeing. So as soon as these variants arrive really in any appreciable number, then they should be known in the community. But on an individual patient, we may not be able to know if it's a variant or not. It's kind of more epidemiology information of what's circulating in the community.
Very interesting. So let's stick with you for another follow-up question that's just kind of related to the massive expansion that you experienced. So you've been at Northwest Pathology since its inception, is that correct?
Well, not entirely. It kind of morphed in different names, started in the 90s. I came in 2006 with Northwest Pathology. And then in the beginning of 2019, we started this additional lab called Northwest Laboratory, which is kind of an offshoot.
Okay. And so in that time that you've been there since 2006, you know, this expansion has been extremely rapid. And we've got all these students here. Can you give us a little bit more insight into the role that Western students have played in this expansion and how they may have helped that?
Yeah, for sure. Yeah, I mean, it was amazing. You know, I mean, like a year ago, we were maybe 100 people, which seemed to me a lot at that time. And now we're close to 500. And I mean, so yeah, it's been jaw-dropping. And a good portion of those folks have roots from Western Washington, you know, through your ability to network and get people interested to join us, and particularly the highly trained people that are actually doing the test are heavily enriched in folks from Western. And they've been a really amazing group of people to work with because they're smart, they're motivated. And during this past year, you know, there were many stressful times. You know, everybody was stressed. You know, particularly, there were several times, particularly in June, when we were doing a lot of testing for Florida. And then right after Thanksgiving were 2 times that we just got hammered because it can be very difficult to control the number of specimens coming in the door because people just start sending you specimens. And so it can be it's not like a faucet that you can turn on and off. And so you try to do as best you can to prepare for high volumes, you know, and then suddenly they come and it's like even more than you even expected. And everyone just rolls up their sleeves, everyone's working together. You know, there were stressful times, you know, like early on, for example, you know, N95 masks were in critical supply. You know, in normal times, you'd never think of asking someone to reuse their N95 mask. It would be unthinkable, but there were no N95 masks. So we had to re-sterilize them with ethanol. And just people's ability to roll with these stressful conditions and realize, hey, we just have a problem. We need to work together and get this solved was really amazing. And so all these folks, and particularly from Western, their can-do attitudes to just join and be willing to help and work together, you know, made it possible and, you know, made this tremendous expansion actually work out.
Very cool. And so before this kind of leads into one of the audience questions that before we get back to the students, somebody in the audience asked, where did that question go? How many tests has Northwest Pathology actually done since the pandemic started? And does the lab really serve the entire country?
Yeah, so we've done, I think, 1.63 million tests now, which is University of Washington crossed the 2 million mark not too long ago. But we're way smaller than the University of Washington, and so that's pretty amazing. And so in the Northwest, I believe we're the second highest capacity lab, next to the University of Washington. And so over 1.6 million tests. And as far as the entire country, well, not from every state, but we've done testing from 40 different states. And so that's 80% of the country, and have done testing for Canada as well. I think we got maybe 1 or 2 tests from Mexico. So I guess technically we could add Mexico, but that was just a couple. But yeah, so 40 different states and 1.6 million tests.
Thank you. So let's get back to the students. And here's a question for the graduate students and Dr. Singh Khande as well. So what are the benefits of the master's degree program here at Western for students such as yourselves that are aiming to work in industry or biotech? Are there any specific aspects of the Western biology program that you think really benefited you in pursuing these career pathways? And let's start off with Hannah on this one.
Yeah, so I think that the master's program at Western was a good choice for me. One of, I think, the best features of it that's translated well into my work here is being able to do investigative research and kind of have a little bit of ownership over it. I have a project that I work at Western, and there's times where that needs to be troubleshooted and things like that. And so that skill set has translated well into this environment. I think it being a master's program, specifically in my case, has been amazing to be able to decide for myself like which offshoot of biology is the right one for me, whether that's more clinical based or research based, and having this experience where I can work on my project, I can have a job at Northwest Pathology, and really let all of that skill set come together, helps me make a better decision on what I want to do with my future. And also the networking opportunities that have come out of this. We work with a lot of, in our master's program, specifically at Western, my cohort they're biologists, and that's amazing. We all get along really well, but working here, we get to work with biochemists, chemists. The Western students that have helped with this experience come from also like outside of the biology department as well. And it's been a really cool experience to get to work with people that I wouldn't normally work with. So yeah, I think these two experiences together has really been beneficial for me to learn how to collaborate across disciplines, problem solve, and get to know some people along the way.
Very nice, thank you. How about yourself, Benjamin? So what are the benefits of the master's program here for people like yourself that are aiming to move on to work in industry or biotech or other related fields?
Sure, yeah, definitely. I really appreciate Western's master's program from a couple different standpoints. Right when you're joining the master's program, probably the first thing you notice is how interdisciplinary and collaborative the culture is, and how supportive all the faculty are to making sure you're achieving your goals that you are specifically looking after. So each student is a specific student and learns in a specific way and is going towards specific goals. And Western really champions our ability to do that. So Western allowing us to have a platform for really taking ownership of our research experiences and our research projects allows us to better be able to to have agency when we jump into a place like Northwest Pathology and are working towards really fine tuning a experiment or a test. And then going in and having opportunities to do a bunch of different opportunities at Western and being supported in unique ways, such as teaching, networking, research, classes, all these things where we get a lot of wonderful mentorship, allows us to have a lot more comfort when we're working on ramping up that testing and ramping up those experiments to start doing things more productively or more wholesale. So going from small scale fine-tune research projects that we do at Western to very high throughput experiments that we do at Northwest Pathology is really helped out by the fact that we're encouraged to be critical thinking and fast thinking and to be leaders of our own experiments here at Western. So I feel like that a lot of the technical skills that I get at Western are useful from the point of view of having a baseline for being able to be productive in labs like Northwest Path or the other biotechnology applications, but also having a culture that's so supportive and so collaborative allows me to better push forward the experiments at Northwest Pathology and have more agency in when I'm trying to translate the skills and the networking that I'm developing in Northwest Pathology or otherwise to long-term career goals. So I think from both the small scale and from the long-term career scale, Western and Northwest Pathology do a great job of allowing you to have agency and confidence when you're pursuing those goals.
Thanks, Benjamin. So Dr. Singh Khande, you've mentored a bunch of graduate students here at Western through the master's degree program. Do you have any kind of anecdotes or examples of how the master's program has set students up, either from your lab or other labs, to succeed in industry or biotech or any of these other fields?
Yes, there are many, many examples. Our graduate students go on to either PhD programs or as independent scientists in government-based labs. My own, two of my 4 recent graduate students are now leaders in the USDA because I'm a plant and molecular biologist. So they've gone into great careers in government-based research and then in private research as well. So yeah, broad. One of the great strengths, the students have articulated the strengths of our graduate program beautifully themselves, and that collaborative nature rather than competitive. We are not cagey about our research projects. We share with each other. Nick, you're a great example of how much Nick reaches out to help people with microscopy related, high-end microscopy training, which is very unusual for a research institution our size, and how sharing Nick is. Nick is just a marvelous example of that. He'd be our poster person for sharing and generosity. And that spirit you can see students have absorbed as well, experienced and benefited from and absorbed and will pass on, I'm sure. So yeah, that collaborative rather than competitive nature of the research philosophy here, and the breadth that students have already mentioned, the interdisciplinary breadth in collaboration with chemistry. Nick, again, you're a great example of collaborations, right? You have a collaboration with the chemistry faculty. So, and students benefit enormously knowing that, understanding that research questions are not siloed, really. The best way to solve problems is by cross-fertilization and by integrating approaches from many different angles and many different types of expertise. So those, I think, are the strengths of our program. And our students do end up with very fine careers in a breadth of both private as well as public institutions.
Well, thank you for that, Anu, and thank you for the shout out. When you have beautiful critters like this thing behind me to image with a microscope, it becomes a lot of fun. So I do appreciate that. But I agree that there's a very strong collaborative environment here, which is very, very unique for a program like this. So I think unfortunately, we're getting close to the end, and there's a bunch of great audience questions. So I'm going to go a little bit kind of off script here, and let's get to some of these really interesting questions from the audience. I'm not always sure who would be the best person to answer these, but I think Dr. Wolgamott might be on the hook for a few of them because they're related to some logistical aspects of the testing. So one person asked, so it looks like you've gone from 100 to 500 employees in the last year, which is pretty remarkable. And now, assuming that there's a post-COVID return to normal, what's the likely number of people size of the firm in the post-COVID era?
Yeah, I wish I knew. So eventually the wave will pass, and probably a lot of people will move on to the next steps in their careers, and hopefully have earn something, good experiences to help take their next step. But there is going to be a tail of testing that will be significant. And so the testing is going to shift. In the past, it's really been individual based tests, predominantly people that are ill, trying to figure out who exactly has it and has not. And now there's going to be a shift into surveillance testing. And so what we're working on now is validating batch testing, which was mentioned, I think maybe Benjamin mentioned it. And our people from Western have participated and helped design the experiments to demonstrate that we can actually do batch tests. So what this means is that you can have a classroom and all 25 people swab their noses and stick them all in one tube. And then if the tube is negative, then everybody's negative. If there's any positives in there, then you have to go back and figure out who's positive. And so that works well in cases where there's a really low prevalence, but you still want to tease it out. And that will be important because even though there's a vaccine now, probably about 30% of the population for some reason or another will not be getting the vaccine. And so I think there's going to be a tail. And a lot of people are saying that COVID testing is going to persist into 2022. And so we'll see if that's true or not, but it is true that the US government is putting forward funding programs and strategies to try to test basically all the schools in the nation on a fairly regular basis to try to find outbreaks and stomp those out early on. And so we'll probably be part of that. And so there will be significant tail of testing that will go on. And then the other thing is we're also trying to, now that we have, a team of really smart people and we've got equipment and such, we're also trying to work up and work more into sexually transmitted infection detection, Neisseria, gonorrhea, chlamydia, HIV, HPV, things like that. And so we'll probably do some more of that, not nearly at the scale as COVID, of course. So to get back to your question, I don't really know what the lab is going to look like a year from now. I think that coming this fall and winter, it'll look fairly similar to the way it looks now. But I imagine eventually that we'll probably taper down to a couple hundred people, and hopefully, as for people that want to stay on with us, hopefully there will be opportunities for them to stay and continue working. And then for other folks, hopefully they've had some good experiences and will for sure write great recommendations for the next steps in their career.
Hey, thank you. And so you kind of touched on another question from the audience. It sounds like somebody was wondering whether or not you're going to be taking part in the pooled sample testing from the sewer systems. And it sounds like in some way or another, you're definitely set up to do pooled testing from a variety of sources.
Yeah. So most of the pooled testing that we're looking into are nasal swabs and such. And we can learn more about sewer testing. I have some concerns about the sensitivity of that, just because any particular, COVID is shed in the stool, by the way. It can be detected, and so it does end up in the sewer. But the problem is it gets so massively diluted, by all the dishwashers and showers and clothes washers with all these different detergents that break open the virus and then cause the RNA to be degraded. That I've got significant concerns about the sensitivity of that, and so we'll be looking into that. And if it's valid, we'll participate. But when I'm talking about pooled testing and batch testing, mostly it's kind of closer to our traditional testing. It's just that you've got a whole bunch of swabs in a single tube that you're testing.
Okay. So it looks like we're probably going to have time for maybe one more question. And so there's a question in the audience that was really posed to anyone, but I'm going to end it with the undergraduate alums. And so it seems like one big change from your lab experiences at Western to working at Northwest Pathology is in terms of the scale. Moving from relatively small scale experiments like what you do in 324 to the much larger high throughput processes. For you as students, were there any challenges to making the transition from kind of the individual experiment that you do in lab to the scale of the experiment that you were working on at Northwest Pathology? And why don't we go with Carrie first?
Yeah, working in a research lab is definitely very different from here. Learning how to work in a high throughput, fast paced environment was a learning curve, but it was fun. So yeah, I think, there's challenges with any new experience, but, you improvise, you adapt, you figure it out. And there's going to be a lot more of that since I just graduated, and hopefully I can keep having new experiences until I figure out what I want to do for my career.
Cool. How about yourself, Aliki? This change in scale, how were you prepared? How did it impact you?
I think just because, like Carrie said, it was such a fast-paced environment. Every time I'd come in, things would be changing. And so there was always that, communication and trying to be on the same page with, 10 other people. In a way, it was very chaotic at the time, but we made it work. I think for me, it was such a surreal experience because, throughout your college degree, you form this community support system. And that same network I created throughout my time at Western ended up working at NWP. So like in the past, it was like, okay, we're here as students, we're learning, we're studying together, doing research together. And then fast forward to when the pandemic started, now we're here together in a real life situation, doing a job that requires all the things we spent so many years studying. So in that way, it was a really interesting dynamic.
Okay. So we wanted to wrap up with everybody giving 20 seconds to just say one thing they've learned. Let's shorten that because we're running out of time. Maybe one word, and I'll start. What's one word that would describe what you've learned since the pandemic started and working at Northwest Pathology? I'm going to start off and I'm going to say the word flexibility. How about yourself, Dr. Wolgamott? One word, what have you learned?
Collaboration.
Collaboration. Dr. Singh Khande?
Inspired, especially inspired by students and their abilities.
Benjamin?
Yeah, probably collaboration or momentum.
Momentum, I like that. Hannah?
It's a tough one. Probably like adaptability.
Gary?
That's what I was gonna say. I was gonna say adaptability.
Perfect. Aliki?
I think confidence.
Confidence. Very cool. All right, so that brings us to the end of our time together, and I'm going to turn it over to Brad now to lead us to the end of the webinar.
All right, thank you very much, Nick. And I want to say a special thank you to all of the panelists. And hopefully those of you watching get a feeling for why I feel such a great sense of pride in serving an organization with such remarkable students and remarkable faculty and staff. It's really a great, I really appreciate everybody. And from all of us at Western Washington University and the College of Science and Engineering, we want to thank you all for joining us today. And we also want to thank the Western Alumni Association for their partnership. And we'd like to thank those of you who chose to support the cell and molecular biology students as part of this webinar. A recording of this webinar will be emailed out in the next few days, so please check your email soon for the link and feel free to share it with others who may not have been able to attend today. So once again, my sincere thank you, and please have a great day.